Hello friends, today we will be studying Histology of Small Intestine Part 1. In this video, we will be doing the general features of Histology of Small Intestine, and we will be studying one important slide, and that is of Histology of Duodenum. Now, small intestine is a 6-meter long mucomuscular tube, and it has three parts: duodenum, jejunum and ileum. Duodenum is the first 25 centimeters and extends from the pyloric end of the stomach to the duodenojejunal junction. Next is the jejunum, which is the middle 2.5 meters, and terminal is the ileum. It's the distal 3.5 meters, and this is what continues with the large intestine at the ileocecal junction. Now let us see the general features of small intestine. Small intestine is a site of digestion and absorption of nutrients. To carry out the same, it has a few modifications that increase the surface area of absorption, and these are plica circularis, villi, microvilli and crypts of Liberkuhn. We had discussed all these four in detail in the video on general features of the gastrointestinal tract. We will still see a few details of each now. Plica circularis or circular folds, also known as valves of Kerckring. These are permanent circular folds that contain core of submucosa. They predominantly are seen in the duodenum, except in the first 5 to 6 centimeters, and in the jejunum, and these are absent in the terminal part of ileum. These folds decrease the speed of passage of the intestinal contents, and they increase the intestinal surface area. And this picture shows us the interior of the jejunum, and here we see these permanent circular folds on the mucosal surface, and those are the plica circularis. Villi. These are projections of lamina propria. Core of villus contains blood vessels and lacteals or lymphatic vessels. They give a velvety appearance to the intestinal surface on gross examination. And this picture here shows us the mucosal projections, which are the villi. Next we see microvilli. Epithelial lining of small intestine shows numerous microvilli, giving rise to the striated border along its luminal surface. What we see here is a diagram of a cell which is seen lining the small intestine, and the luminal surface shows number of microvilli, and this is what gives rise to the striated border-like appearance. Crypts of Liberkuhn. These are simple tubular intestinal glands that extend in the lamina propria and go up to the muscularis mucosae. They are lined by tall columnar epithelium that is continuous with the epithelial lining of the villi. And this diagram here shows us the villi and the crypts. As we see here, the crypts occupy the lamina propria and extend up to the muscularis mucosae. Now we go on to see the cells of the small intestine. Small intestine is lined by simple columnar epithelium. This epithelium consists of 5 types of cells: enterocytes, goblet cells, Paneth cells, enteroendocrine cells, and M cells. Enterocytes are absorptive columnar cells. These are tall columnar cells with a basal oval nucleus. The apical or the luminal surface of the cell has a striated border formed by regularly arranged microvilli. and each microvillus has a core of vertical microfilaments covered by plasma membrane. The function of the enterocyte is that the cells absorb nutrients and they secrete enzymes required for terminal digestion. This here is a diagram showing a single enterocyte, tall columnar cell with an oval basal nucleus and the luminal surface showing regularly arranged microvilli. Next we study the goblet cells. These are unicellular glands that secrete mucus. The number of goblet cells increases gradually from duodenum towards terminal part of ileum. The cell is shaped like a drinking glass, hence called a goblet, broad above and having a narrow stem towards the basement membrane. The apical portion of the cell contains mucinogen granules. These granules are washed away during tissue processing and H&E staining, hence the apical portion looks empty in an H&E stained slide. The nucleus is pushed towards the base and becomes flattened. What we see here is a diagram showing us a goblet cell, broad near the luminal surface and a narrow stem towards the basement membrane. We see mucinogen granules in the apical portion and we see a flattened nucleus which is pushed towards the basement membrane. Paneth cells or zymogen cells. These are present in clusters at the base of the intestinal glands. Cells have intensely acidophilic refractory secretory vesicles or granules in the apical portion. Nucleus is oval and basally placed. And this here is a diagram showing us the Paneth cell. Oval nucleus placed towards the base of the cell or near the basement membrane, and acidophilic vesicles or granules near the apical portion. Function of Paneth cells: secretion of zymogen granules containing lysozymes, alpha defensins, cryptidines, TNF-alpha and zinc. Lysozyme and alpha defensins help in controlling the normal bacterial flora of the small intestine and thereby maintain the innate immunity of the crypts. The Paneth cells can phagocytose bacteria and their lifespan is 30 days. Enteroendocrine cells or argentaphine cells. Few cells are present among the epithelial cells of intestine. They produce both different peptide hormones like enkephalins, vasoactive inhibitory peptide or VIP. There are two types of enteroendocrine cells, closed type and open type. In closed type, the cells do not reach the lumen of the gland or the crypt, while in the open type, the cells reach the lumen of the gland. This here is a diagram showing us the two types of enteroendocrine cells. This is a closed type of cell where you can see that the cell does not reach the luminal surface, while this is an open type of enteroendocrine cell. M cells, epithelial cells that overlie Peyer's patches and other intestinal lymphatic follicles. Cells have deep pocket-like recesses or spaces that accommodate lymphocytes, macrophages, and dendritic cells. They have few microfolds on their apical surface. They do not have any microvilli. Cells have protein receptors that catch bacteria and transfer bacterial proteins to the macrophages. Thus, M cells act as antigen transporting cells. And this here is how an M cell looks like. And what we see here is a pocket. intermediate or undifferentiated cells. These behave like stem cells and give rise to other intestinal epithelial cells. They are present in the lower half of the intestinal gland or crypts of Lieberkühn. These are columnar cells with short, irregular microvilli and some apical cytoplasm. Now we go on to study the duodenum. This is the proximal 25 centimeters or 10 inches of the small intestine. It receives food material from the stomach, which is highly acidic. It also receives secretions of the liver, that is the bile, and the pancreas, that is the pancreatic juice, through common bile duct and accessory pancreatic duct into the second part of duodenum. Pancreatic juice is alkaline and neutralizes the acidic content of the stomach in the duodenum. Now we go on to study the important slide, that is histology of duodenum. Wall of duodenum consists of 4 layers from within outwards: mucosa, submucosa, muscularis externa, and serosa or adventitia. This here is a diagram showing the histology of duodenum. What we see here is the luminal surface, and this here is the mucosa made up of lining epithelium, lamina propria, and muscularis mucosae. Next is the submucosa. Then comes the muscularis externa, having the 2 layers, inner circular and outer longitudinally arranged smooth muscle fibers, and outermost is the serosa or adventitia. Mucosa. Mucosa shows plicae circularis. These are permanent circular folds having a core of submucosa. Villi. These are finger-like mucosal projections having a core of lamina propria. Villi here are tongue-shaped and contain fenestrated capillaries, blind end lacteals or lymphatics, and smooth muscle cells. Epithelium. Villi are lined by simple columnar epithelium with few goblet cells. Microvilli. Luminal surface of columnar epithelial cells shows striated border due to regularly placed microvilli. This is the same diagram where we see the lining epithelium. The lining epithelium is made up of simple columnar epithelium, and in between them we see few goblet cells. And what we see here, the mucosal projections that contain the lamina propria as core, these are the villi. Intestinal glands or crypts of Lieberkühn. Short tubular glands formed by invagination of bases of crypts in lamina propria. Glands are lined by columnar cells, goblet cells, Paneth cells, and enteroendocrine cells. In this diagram, what we see here is the crypts of Lieberkühn. Next we see the muscularis mucosae. Has 2 layers, inner circular and outer longitudinal layers of smooth muscle cells. This what we see here in the lowermost part or the deepest part of the mucosa is muscularis mucosae, the inner circular and outer longitudinal layer of smooth muscle fibers. Thus we have studied the mucosa, which is made up of lining epithelium, lamina propria and muscularis mucosae. Next we go on to see the submucosa. Brunner's glands, highly branched tubulo-acinar mucus secreting glands seen in the submucosa of duodenum. Acini are lined by simple cuboidal or low columnar pyramidal cells. Cells have an empty cytoplasm and a basal flat nucleus typical of a mucus acinus. Ducts of these glands pass through the muscularis mucosae and open into the bases of the villi. These glands occupy most of the submucosa, and their function is that they secrete mucus that protects the duodenum from the acidic contents of the stomach. This here we see the submucosa, and as is seen here, it is completely occupied by the Brunner's glands, and these are seen as typical mucus acini lined by cuboidal or columnar pyramidal cells having a flattened nucleus towards the base of the cell and an empty or a clear cytoplasm. Next we see the muscularis externa. This has two layers, inner circular and outer longitudinal smooth muscle layers, and outermost is the serosa or adventitia. Most of the duodenum is covered by connective tissue, hence is called as having adventitia, except a small part, that is the first 5 to 6 centimeters, that is covered by peritoneum and is called as the serosa. This is made up of loose connective tissue which contains blood vessels, adipocytes and fibers. This here we see the muscularis externa, made up of inner circular and outer longitudinal layer of smooth muscle fibers, and this here outermost is the serosa or adventitia, containing loose connective tissue, blood vessels, adipocytes and some other cells and fibers. What we see here is a scanner view of histology of duodenum. What we see here is the mucosa, where we see villi, mucosal projections, tongue-shaped villi, and the lower part we see the crypts of Lieberkühn. What we see here is the muscularis mucosae. Next we see the submucosa, which has got the Brunner's glands. These are the mucus acini that are seen here. Next we see the muscularis externa, inner circular and outer longitudinal layer of smooth muscle fibers, and outermost is the serosa or adventitia. This is a low power field view of histology of duodenum. What we see here is the mucosa, tongue-shaped villi. In the lower part we see the crypts of Lieberkühn, muscularis mucosae, and submucosa showing the Brunner's glands. Another low power view showing us the characteristic tongue-shaped villi, crypts of Lieberkühn, muscularis mucosae, and submucosa filled with mucus acini, and those are the Brunner's glands. So two characteristic features for identifying a slide of duodenum: tongue-shaped villi and Brunner's glands in the submucosa. A high power view showing us the Brunner's glands. This is the submucosa containing the Brunner's glands, mucus acini having a clear cytoplasm and a flattened nucleus towards the base of the cells. This is an epirioimage picture of histology of duodenum. What we see here is the luminal surface. We see these mucosal projections. Those are the tongue-shaped villi. Next we see the crypts of Lieberkühn in the lower part of the mucosa. The lowermost part of mucosa we see the muscularis mucosae. Next is the submucosa, which shows presence of mucus type of acini, and those are the Brunner's glands. Next we see the muscularis externa, inner circular and outer longitudinal layer, and outermost we see the serosa or adventitia. The salient features for spotting for a slide of histology of duodenum transverse section will be presence of numerous tongue-shaped villi, presence of crypts of Lieberkühn, and presence of Brunner's glands in submucosa. So we have studied in this video the general features of small intestine, the cells seen in the lining epithelium of small intestine, and one important slide of histology of duodenum is what we studied in this video. In the next video, we will be studying the other two important slides, that is of histology of jejunum and histology of ileum. And finally, we will see how to differentiate between the three slides of small intestine from each other. For further reference, you can read Sontakke Yogesh, Textbook of Human Histology by CBS Publishers and Distributors Private Limited, New Delhi, 2020, page numbers 209 to 214. Thank you.